So this section of your EOC study guide is going to look at DNA and protein synthesis. If you get your handbook out and your notes out, this will be on page, starts on page 49 is where the notes, oh no, even a little further back. It's going to start on page 43. So this section of notes on DNA is going to start on page 43. So I would actually encourage you to pause the video, stop, and read your notes, pages 43, so that you can remember this material before you try to fill out the study guide. But if you're ready to keep going, you should be able to recognize this picture. Now it's sideways, so if you tilt your head, what you'll see are these letters A, G, C, C, A, G. And what we'll see are phosphates and sugars, and so we have this backbone. And so the black pentagons are the sugars. This is DNA, so this is deoxyribose sugars. And it says draw an arrow to the nitrogen bases. Each of these is a nitrogen base. Adenine is a nitrogen base. Guanine, cytosine, adenine, guanine. All of these are nitrogen bases. It also wants the nucleotide sequence that would be on the complementary strand. Remember, it means they go together. So what would go with A? Well, maybe let's start at this end, and we'll read left to right. What would go with G? Well, remember our DNA, let's write this together. Our DNA base pairs are A with T and G with C. So if I have a G, it would base pair with a C. If I have an A, it would base pair with a T. So you should be able to finish base pairing to make the complementary strand. So this is the first section that looks at what is the structure of DNA. If the strand of DNA above undergoes transcription, now be careful because we have three big processes. Replication, which makes more DNA. Transcription, which makes mRNA, and translation, which brings the amino acids together. So if the strand of DNA underwent transcription, what would the mRNA be? Notice that we write it in sets of 3. Now, RNA, remember, our RNA base pairs are A with U and G with C. And so what would go with C, or with G would still be C, but this time what would go with A would actually be U. And what would base pair with C would actually be G, C-U-G. Now remember, that's called a codon. Those are the first 3 nucleotides of the mRNA, and they're called a codon. So there's your answer to what is a codon. I just said it, so if you didn't hear it, pause the video and rewind it. You should be able to use the mRNA in this table to find C-U-G. We'll do the first one together. First letter is C, so I know I'm somewhere here. Second letter is U, so that means I'm in this box. And then my third letter is G, so I need to choose this bottom one, L-E-U, and so that's leucine. Next, it asks us to compare RNA and DNA in the following table. And so it's asking us to compare the sugar, the bases, the number of strands, where in the cell, and the function. What we're going to do together is the function. The function of DNA is to contain the genetic info for building proteins and to pass it to the next generation. So DNA has 2 jobs. One, it holds the genetic information. Number 2, it passes that information to the next generation of cells or of living organisms. The function of RNA is to use the DNA code to build a protein. Protein. That got a little sloppy there. Protein. Use the DNA code to build a protein. Now, RNA and DNA do have different sugars, different bases. Remember, the DNA bases are A with T and G with C. They do have a different number of strands. Remember, RNA is always single-stranded, DNA is always double-stranded. The DNA in a eukaryote is in the nucleus, but keep in mind that prokaryotes don't have a nucleus. The RNA might be in the nucleus, and and it might be in the cytoplasm. So you should be able to fill out the rest of this box by going back to read your notes. The next question, what kind of bonds hold amino acids together? So be careful, because if I have A across from T, those are what we call weak hydrogen bonds. But it's asking about what holds amino acids together. This is actually a bond that starts with a PE. I'll leave it there. But this type of bond is much stronger, and that's what holds amino acids together. The next section is really going to dive deep into DNA, mRNA, tRNA, and amino acids. And so in this box, it wants to know what are the functions of DNA, mRNA, tRNA, and amino acids. And so we just did the function of DNA. The function of mRNA is to let's not use that. Let's say have a base pair of the gene to leave the nucleus. tRNA is to deliver the amino acids, and proteins do the work in your body. So DNA has the code for proteins, and then this is how we build it. This next section talks about mutations. Remember, there are two types of mutations. There are what are called substitution mutations, and then there are frame shift mutations. Transcription occurs in the blank of the cell and makes a copy of blank from DNA. So it's going to make a copy of a gene from the DNA. It doesn't copy the whole DNA, just one section called a gene. The mRNA leaves the nucleus and goes to V, starts with a C, to bind to A, starts with an R. The anticodon, let's underline the T in anticodon. The codon is on the mRNA, the anticodon is on another type of RNA. This molecule has N. This is a two-word phrase, AA, attached to it. Amino acids are linked together to create A, starts with AP. True or false, all of an organism's cells have the exact same DNA. It says explain how you know this is true. Well, it is true because they inherited it. They inherited, let's say, copies from the first cell. But we do need to put that different sections or genes are used in each cell. Now, one thing that wasn't talked about in this section is DNA replication. So definitely make sure that although this is a long study guide with a lot of information, it doesn't include everything. So make sure that you have read those notes about DNA structure and DNA replication. The next section is on cell specialization, which is what we were just talking about. This idea that different cells use different parts of the genes. How does the structure allow it to form its job? Well, the structure of the red blood cell is that it has a hollow center with hemoglobin. Hemoglobin is a protein that allows it to carry oxygen. The nerve cell is really long and slender, and this allows it to send messages quickly to help with regulation. Remember, the animal body regulates its movements, regulates how the animal responds to help with regulating or controlling the body. Finally, we have the muscle cell. The muscle cell is going to have a large surface area, and this is going to help it to get a lot of energy. Inside of the muscle cell are actually going to be hundreds of this organelle. So you should be able to recognize this organelle by its picture. The last section that we'll talk about here is going to be biotechnology. What is the purpose of the Human Genome Project? This question almost always shows up on the EOC, so we really want to make sure that we go back and read our notes on biotechnology. If you're looking for where the notes on biotechnology are, let me find them for you. These notes on biotechnology are going to be on page 65 through 70. 65 through 70. The purpose of the Human Genome Project is actually to find the location of human genes so that we might one day be able to treat or cure diseases. Cloning is where we make exact copies, so we are able to make exact copies of an organism. The process that makes a DNA fingerprint, this is exactly in your notes, so please make sure that you go back to do that. It's a two-word process. One of them starts with a G, and the second word starts with an E. Look at the DNA fingerprint to the right, which individuals are most closely related, so you should be able to do that. Number 5, remember the number one most frequently asked question about the use of genetic engineering is to make insulin or to replace missing hormones or body chemicals. So in this next picture, we're looking at genetic engineering, and we're looking at the steps, and it wants you to describe what is happening. The first step, in step 1, we'll label that number 1, is find and cleave, which means to cut the desired gene. Step 2, which is down there, is to cleave the vector. Now remember, to do this, we have to use restriction enzymes. So make sure that you're studying that word, restriction enzymes. We used it as a note card. Number 3 is to splice or combine them together. Combine the desired gene and combine the vehicle. And step 4 is to put it into the host cell. So insert into the host. Now keep in mind that most of the hosts are going to be bacterial cells because they are so easy to manipulate. We are going to try to power through this next section on mitosis and meiosis as well. So we're going to move a little bit quicker through this section because this is directly in your notes. Your notes on mitosis and meiosis are going to be back in your notes a little bit. They're going to start on page 35. So these notes are going to start on page 35. If you read through your notes, you should be able to get through this pretty quickly. One important thing is that mitosis divides once and meiosis divides twice. Remember, mitosis makes 2 cells. So meiosis, you don't have to memorize everything here. If you study one, you'll remember the other. Meiosis actually makes the other one, which is 4. The chromosome number of the mother cell in both of them starts as diploid. But remember, meiosis makes sex cells, which are going to be haploid, and mitosis makes identical cells, which are going to be identical to the parent, or diploid. Remember, meiosis prepares, so it's part of sexual reproduction. When does replication happen? For both of them, it's during the S phase of interphase. Writing a little bit sloppy here just because I'm trying to get through this quickly. Both of them happen during the S phase of interphase. Remember, this one makes identical cells, so if we started with 50 chromosomes, we should end with 50 chromosomes. These are making haploid cells, so if we start with 50, we should end with 25. That's the bell to come back from lunch. I'm filming the video during lunch today. You should be able to find and put these cells in the correct order, and then you should be able to write a sentence using these words that puts them in order. So that's your video today. Covered a lot, but make sure that you take the time to read over this.